Hello, biologists. I wanted to give you guys some content today regarding RNA and give you a quick introduction to this idea called central dogma. So today we're going to compare and contrast the structural and functional differences between RNA and DNA, and then we're going to talk about the process of protein synthesis and ultimately how proteins are made in the cell. One of the steps involves making RNA as part of that process. So let's start by comparing RNA and DNA. So we see that there are two different types of nucleic acids pictured here. We have DNA and RNA. They're named differently because they contain different sugars that make up their backbone. Remember, the backbone is the sugar phosphate backbone, where we have sugars alternatingly bonding with phosphate groups. The sugar that makes up the backbone of RNA molecules is called ribose, which is shown in its name, right? Ribonucleic acid, whereas a different type of sugar makes up the backbone for DNA molecules. So the sugar that makes up these backbones, shown here in blue, are deoxyribose, a different type of 5-carbon sugar. So we reflect that difference in the names of these molecules. DNA is an abbreviation for deoxyribonucleic acid, and RNA is our abbreviation for ribonucleic acid. Another difference, which is pretty obvious, is the fact that RNA is usually single-stranded, whereas DNA is our double-stranded molecule. We see that this is a double helix. We can disrupt these two strands by disrupting the hydrogen bonds that hold these two strands together, and that's going to be helpful if the cell is undergoing DNA replication or transcription. But RNA we usually see as a single-stranded molecule. Another point I want to make about the structural similarities and differences is that some of the nitrogenous bases we can find in both of these molecules, but some are unique to only one of these molecules. So we can see guanine, cytosine, and adenine are nitrogenous bases that are found in both RNA and DNA molecules. When we look at the ones that are listed down here, these are specific to RNA, and this one's specific to DNA. So it's only in DNA molecules that we'll find this nitrogenous base, thymine, and it's only in RNA that we'll find this nitrogenous base, uracil. So both of these base paired to adenine. So uracil will match up with adenine, as does thymine, but we won't see any thymine when we're making the complementary base pair to adenine when we're building our RNA molecule. So T's are essentially replaced with U's. So let's talk quickly, just an overview about this idea of central dogma. This is the overview of how we take information that's stored in your genome or your DNA to express certain traits. So we've talked about genetic traits, your eye color, your hair color, your ability to taste certain things. Those are all traits that have to do with what's stored in your genetic code. And those traits are expressed by making proteins. So we take this DNA sequence, and that sequence is then used to make the protein by first making a molecule of RNA, and that RNA is then used to make the protein. So this essentially is a two-step process, and the first step is called transcription. And this happens in the nucleus of cells because remember, that's where DNA is stored. So we're using the DNA that's stored in the nucleus, and it contains this sequence that we're going to need to use to make this protein. But since the DNA has to stay in the nucleus, we have to make a transcript or kind of a copy of this information, and we do that by building a molecule of RNA here. And this RNA molecule is a product of this process called transcription. Eventually, this RNA molecule will leave the nucleus, go out into the cytoplasm, where the rest of protein synthesis can occur. That process is called translation.